Good evening all, good evening, good evening. Good evening, good evening, good evening all, good evening. Okay, let's start. So friends, today I am taking cell signaling, and once again, not tomorrow, day after tomorrow, once again I will take the cell signaling and cell communication. Till Sunday I will be continued, and madam will come on Monday, and Tuesday she will finish the left topics of the cell biology. Once again immediately I'll continue with the next topics. Okay, yes, let's continue. Okay, this class is up to 45 minutes. Please continue, friends. Okay. Now let's understand cell signaling. Okay. Is there any necessity that we have to study the cell signaling? Is it compulsory? Of course, friends, the cell signaling is compulsory. Why? Because we all know very well we can expect the question from the cell signaling in part B also. Good evening all friends, even we can expect the question in the part C also. There's no doubt about it. Hence my friends, it's compulsory and necessary that we have to read the cell signaling topic without fail. And I always request you, in cell signaling topic friends, I want you to apply your knowledge. When you apply your knowledge, however the difficulty, whatever the way they may ask the question, whether it's a factual statement, experimental based, analytical based question, you can answer it very easily. So please kindly do not worry about that. Okay, for that try to understand the depth of the concept. Okay, let's start. So cell signaling. Okay, friends, everyone try to understand. Do you think cell signaling is an important process that has to be occurred in the cell? Is it an essential process or it is a necessary process? Of course, friends, in a simple way I can tell you, whatever the function that is occurring in the living cell, every function required the cell signaling. Please kindly do not forget that point at all. In a simple way, if the cell wants to survive, it requires signaling. If the cell wants to divide, it requires the signaling. If the cell wants to differentiate, it requires the signaling. Even my friends, if the cell wants to die, that is the apoptosis or a programmed cell death, it requires the signaling. So what is the statement that you have to understand here? Every single process that is taking place inside the cell, compulsory it requires the signaling. Never forget that. Okay, so friends, what are the various components of the signaling? Everyone listen. What are the types of cell signaling? We all know that there are different types of cell signaling are there. Among those, the first and most important, paracrine signaling, synaptic signaling, autocrine signaling, then friends, endocrine signaling, then what we can say, cell to cell contact signaling. You can also call it as a contact dependent signaling. My friends, I want every one of you look into this beautiful picture. You are able to understand very clearly what is this endocrine signaling. Everyone look into this. Friends, this is a signal releasing cell. This is what? Signal accepting cell. We read, good evening Sumaya. We read many times, if a particular cell want to receive the signal, what it required actually? I told you many times, without receptor, anyway the ligand molecule or a signal molecule cannot attach. That's a common point. Now, why we call it as a endocrine signaling here, and what kind of a ligand molecule or what kind of a signal molecule follows the endocrine signaling? You all know very well. The endocrine signaling can be extremely followed by which cell, friends? By the hormones. We all commonly know, right? Hormones can travel through what? Hormones can travel. See, this is the signal releasing cell. See, this is the cell releasing the hormone. That hormone travel through the bloodstream. That hormone travel through the bloodstream, and where it reaches, friends, it is going to reach the target cell. Where it is going to reach the, it is going to reach the target cell. So in a simple way, the signaling that is happening through the blood stream, we call it as a endocrine signaling, simple way. And friends, what is the paracrine signaling, you are only able to see here the cell only target the nearby cell instead of targeting the long distance cell. You only try to understand. This is the signaling cell. Now this cell released the signal. Do you think this signal will travel through the longer distance? No, not at all. With the help of certain mediator molecule, this particular signal will come and attaches to what, my friends? Targeted cell. The point every one of you has to remember here, what is that point? This signal, this targeted cell is not present in the longer distance. This cell is present at the very nearby distance. In a simple way I can tell, the signaling which is occurring between the two nearby cells, very nearby cells, we call paracrine signaling, and this particular signaling can occur through the chemical mediators. Very simple point. Next one, what do we call it as? Autocrine signaling. What did you understand by the word autocrine signaling here? You all commonly know that autocrine signaling means what? This is the cell released the signal. There is no doubt about it. Friends, the point is very clear. Once this particular signal got released, what is going to happen? Once again this signal will come and attaches to the same cell. That is what do we call autocrine signaling. The best example what you can take a best example of cancer cell. You all commonly know, friends, if the cancer cell wants to grow, survive, develop, what the cancer cell is required? It requires continuous growth factors. Now what happening here? Cancer cell only released this particular growth factors. Now my friends, the point is very clear. That growth factor will come and associated with that particular cell itself, because on this particular cancer cell only there are receptors are there. To that receptors only this will come and attaches. This is what exactly we call, my friends, autocrine signaling. Last and final type, you can see. Good evening Deepika, continue. Sumaya, continue. What do we call the contact dependent signaling? My friends, what did you understand by the word contact dependent signaling? This is the signaling cell and this is the targeted cell. My friends, how does this particular contact dependent signaling is happening? These two signalings are happening by the direct physical contact. By direct physical contact. That is what do we call contact dependent signaling. If you recall our previous lessons, what is the best example we can take? You can take a best example of T helper cell and the macrophage. Macrophage, on its membrane, what they contain? MHC molecule. On the MHC molecule, what is present? Antigen is present. T helper cell having what kind of receptor? CD4 and TCR receptor, T cell receptor. So you only try to understand, T helper cell will come very close contact with the macrophages. With the help of MHC, it will take the antigen. Now what I can call this, friends? I can call this as contact dependent signaling. Never ever forget this particular point at all. So friends, don't forget, what are the four major type of signaling? Please remember all these. Endocrine signaling, paracrine signaling, autocrine signaling. Friends, then followed by what kind of a signaling? Contact dependent signaling. Please don't forget this particular statement at all. Okay, now listen to me carefully. Friends, I was continuously repeatedly saying to all of you that the signal will be released from the signaling cell. Now the point is very clear. If this, but listen my statements very carefully. If this particular signal wants to act on the targeted cell, how it is going to act on the targeted cell? That is the question. If the signal wants to act on the targeted cell, compulsory, there is no doubt about it, it has to attach to the receptor. Without attaching to the receptor, there is no way none of the signaling molecule is going to act on. So friends, listen everyone, what are the types of receptor? There are different type of receptors are there. You can ask me a question, based on what we have categorized the receptor. Everyone listen. Based on what we have categorized the receptor. We have categorized the receptor Quorum sensing, it is a Sumaya, it's a bacterial signaling, right dear? Quorum sensing, we can say it's a kind of autocrine, but not completely, dear. It's a kind of autocrine, but not completely. Okay, but you can say best example is dextracrine. Correct, Deepika. Anyway, listen. Receptor, what kind of receptors can be used by the signaling molecule, or based on what we categorize the receptor? We categorize the receptor into two types actually. What are the two types of receptors? One we can call it as a surface receptor, cell surface receptor. Another one we call it as what, my friends? Nuclear receptor. Okay, now you can ask me a question, sir, what kind of ligand molecule will use as surface receptor? Everyone has to remember, my friends, especially hydrophilic signaling molecule. Only hydrophilic signaling molecule uses this particular cell surface receptor. What kind of ligand molecules are, what kind of signal molecule uses the nuclear receptor, friends? Undoubtedly, my friends, hydrophobic signaling molecule. They are the one which is basically going to utilize the nuclear receptor. Please kindly don't forget the statement. Hydrophilic signaling. What do you mean by the word hydrophilic signaling molecule? Which cannot cross the plasma membrane. Whichever cannot cross the plasma membrane, they can use cell surface receptor. Whichever can cross the cell membrane, they are going to use what? Nuclear receptor. The point is very clear. Is there any necessity compulsory to use the receptor? Without using the receptor, signal molecule can't show their action. It's highly impossible. Without using signal molecule, we cannot use, I mean to say, the signal molecule cannot show their function. They compulsory has to attach us to the receptor. Okay, now let me tell you, friends, what are the examples of cell surface receptor? What are the examples of nuclear receptors? So listen, there are total 4 major classes of receptor. Actually 3. First one is cell surface receptor, ion channel linked receptor, enzyme linked receptor. There are total 3 major classes are there: enzyme linked, ion channel, cell surface. Now what this particular G protein coupled receptor comes under? Of course, friends, G protein coupled receptors are also comes under the cell surface receptors only. My friends, what is the point? You only try to understand why I am using a word cell surface receptor. I have used a word cell surface receptor because it is present on the surface of the cell, present on the cell membrane. So cell surface receptors also known as transmembrane receptor, where they are present on the cell surface. My friends, what are receptors actually? You will take an example, my friends. Take an example. This is a cell surface receptor which is a GPCR. What we can call it as, you are able to understand, it is present within the membrane. Now what I can use a word, every cell surface receptor is a integral protein. What, friends? Every cell surface receptor is a integral protein. So to that particular cell surface receptor, a particular ligand molecule will come and attaches and show their action. Now what is the best example of the cell surface receptor? The best example, or remember the statement without fail, the largest class. What, friends? The largest class of the cell surface receptor. What is that, friends? Z protein coupled receptor. Very simple point. Next one, enzyme linked receptor. These two, even ion channel linked receptor, also a kind of cell surface only, kind of cell surface receptor only. Now I want every one of you pay attention towards this picture. You only look into the picture, ion channel linked receptor. These are all what? Ion channel. My friends, please listen my statements carefully. Here in this picture you are able to see ion channels are always closed. What is the major function of this particular ion channel? Ion channels are open and closed with respect to what? A particular ligand molecule comes and attaches. Whenever a ligand molecule attaches, then only ion channel linked receptor is going to get opened. Otherwise, they always closed only, they never get opened. Okay, fine, the point is very clear. They open. What happens when they open? Once they open, they will be involved in the movement of, what they will be involved in? They will be involved in the movement of ion or molecule across the membrane. In a simple way, you can remember ion channel linked receptors or the cell surface receptor, which are responsible for the movement of the ion and molecule across the membrane. And one more important point is what? Ion channel linked ion channel linked receptors are always closed. When they are going to get opened, they are going to get opened whenever a particular ligand will come and attaches only. Upon attachment of the ligand, the closed receptor get opened, then the ions are going to enter inside. Now you can ask me a question, sir, what kind of ligand can come and open this? I can give you a best example, most of the neurotransmitters. Most of the neurotransmitters will come and open this particular channels. Never ever forget the statement at all. Next one, a G protein linked receptor. What you people has to understand by this Z protein linked receptor? Once again, listen to me carefully. Friends, understand what I am talking. Z protein coupled receptor, we're gonna read one by one elaboratively. It is the largest class of cell surface receptor. And first statement every one of you has to remember for the exam. See, most of the students will read very depth of the G protein coupled receptor, and every student will forget why we will use the G protein coupled receptor. That you have to remember. Means what kind of signal molecule uses G protein coupled receptor? That is the question you have to remember. Mostly Z protein coupled receptors can be used by hormones. Mostly Z protein coupled receptors can be used by the neurotransmitters. Hormones and neurotransmitters majorly uses this particular GPCR. What is the structure of GPCR? We will understand in the upcoming slides elaboratively. Now, so the first class of the first example of the cell surface receptor is ion channel linked receptor. The second class is G protein linked receptor. What is the third class? The third class is enzyme linked receptor. Now everyone listen to me very carefully. Listen to me carefully. This enzyme linked receptor, once again you have to remember, they are also cell surface only. There is no doubt about it. Now the question is very clear, which is important for our exam. What kind of ligand molecules mostly uses this particular enzyme linked receptor? We all know very well, friends, whatever the growth factor present in our body, every single growth factor, every single growth factor will uses this enzyme linked receptor. Every single, we will read it in a table. Example, epidermal growth factor, platelet derived growth factor, insulin growth factor, nerve growth factor. All these growth factors are going to use this particular enzyme linked receptors only. Please kindly do not forget this particular statement at all. Now here comes another very unique point. What is that? Everyone listen to me. This is an enzyme linked receptor having 3 domains. Once again I will explain you. It is an overview I am giving you here. It contains an extracellular domain, it contains a transmembrane domain, and it contains a cytosolic domain. What you can say? An extracellular domain, a transmembrane domain, a cytosolic domain. My friends, you know in the picture it is seen. See here, everyone listen. All the time enzyme linked receptors are like this, exactly like this. Right now see here, they are not attached with each other. But when they associated with each other, that is the point. Whenever a ligand comes and attaches, the moment the ligand comes and attaches, then only they will be associated with each other. Then only they are going to form a dimeric structure. Otherwise they don't form the dimer at all. The moment, everyone listen my statement, the moment they form the dimer, Which domain is basically going to get activated? The cytosolic domain is going to get activated. My friends, the moment a cytosolic domain get activated, immediately what is going to happen? It will undergo the autophosphorylation. Now you can ask me a question, sir, what is the necessity of the autophosphorylation? Very important. If the signaling want to be continued, if the signal transduction wants to occur, if the signal transduction wants to occur, this process should occur compulsory. Never ever forget this particular statement at all. Okay, now listen to me very carefully. So we will talk about one by one elaboratively now. So let's start with the largest class of the cell surface receptor. What is that particular largest class, friends? We call it as a G protein linked receptor. Okay. Why we are calling it as a largest, you can ask me. Why? Because it is more diversified. In where? In our body. Now here comes a very unique statement. If you ask me, sir, what is the major essential processes occur in our body? Listen, what are the various essential processes occur in our body by using GPCR? Then I can tell you a very important one, sensation. Right, and another very important, vision. I can give you touch. These are all essential processes actually takes place in our body or in our cell with the help of GPCR only. Never ever forget that particular point at all. Friends, we all know very well GPCR. What do you mean by the G here? Everyone will forget. G represents guanine. Guanine protein coupled receptor. We called it as a GPCR. Now everyone please kindly look into the picture very carefully here. If you look into the structure of a GPCR, we all know, right? GPCR can transpan the plasma membrane 7 times. Since it is transpanning the 7 times, that's why we can call it as a serpentine receptor. Anyway, every student will remember it is transpanning 7 times. That's an easy point. I want you people to remember when it is transpanning 7 times. See here, example, this is 1 time, 2nd time, 3rd time, 4th time. You know, right, friends, if it is transpanning once, you have to remember, friends, nearly 19 hydrophobic amino acids are required. Once again, listen my statements very carefully. In order to transpan once on the membrane, only once, 19 hydrophobic interactions are required, or 19 hydrophobic amino acids are involved. Now the point is very clear. So it is transpanning 7 times. They can ask you a very simple question which no one can answer. What is the question? They can ask you a question that how many number of amino, listen the question carefully. How many number of amino acids are involved in the 7 transmembrane of the GPCR? So you have to remember 7 into 19. Always remember like this. So in order to transpan 1 time, 19 hydrophobic amino acids are involved. Then in order to transpan 7 times, 7 into 19. Don't forget this point at all. Okay. Now since what receptor the GPCR? We know that the GPCR is a cell surface receptor. What are the various kinds of domains that it is having? My friends, look into this. It is having extracellular domain. We can also call it as ligand binding domain. Next one, we can call it as what, friends? What is this we call it as? We called it as transmembrane domain. And what is this final domain we called? Cytosolic domain. Total 3 domains are there. Extracellular, transmembrane domain, my friends, followed by the cytosolic domain. Such a simple point it is. Now, every one of you listen my statements carefully. Especially the cytosolic domain. I am talking only cytosolic domain, not any other domain. This particular cytosolic domain of the GPCR having a binding site. Which domain, friends? Cytosolic domain having a binding site. To that binding site, who will come and attach us? To this, a specialized protein will come and attach us. What do we call that particular specialized protein? We called it as, my friends, G protein. What do we call it as? We call it as a G protein. Never ever forget this particular point at all. Now here comes another very unique point. The G protein, it's a trimeric protein. It is a heterotrimeric, it's not a homotrimeric. Why heterotrimeric? Because it contains different subunit alpha, beta, gamma. Now once again the question is very clear. Just now I told you that, see here, you are able to see in this picture. The cytosolic domain of the GPCR, we discussed that it is associated with the G protein. Now the question is, which part of the G protein is associated? It is associated always with the alpha part of the GPCR only. Never ever forget this particular statement at all. Okay, it is associated with the alpha part of the GPCR. Now once again everyone remember, this G protein alpha having two binding site. One binding site we can call GDP binding site, another one we can call it as a GTP binding site. We all know very well, we read many times, whenever G protein alpha is in the GDP bounded form, what I can say? I can say it is inactive form. Whenever the G protein alpha it is in the GTP bounded form, what I can say? Active form. Now you can ask me a question, when the G protein alpha will be in the GDP form, when the G protein alpha in the GTP form. Listen friends, whenever there is a presence of ligand or whenever there is a presence of signal molecule. Now you have to remember, if I want to activate the G protein, it is compulsory, it is necessary, it is mandatory. Firstly, the receptor has to be activated. This is a unique statement, my friends. Which has to be activated? Receptor to be activated. How the receptor get activated? Upon ligand binding. Friends, everyone listen. Okay, now finally G protein get activated. Imagine what this point, no one should forget this. What is the point? What is the ultimate goal of the G protein? This is important point. My friends, the ultimate goal of the G protein is, everyone listen to me, activating an important molecule. What do we call it? Effector molecule. Now you can ask me why the effector molecule has to be activated. Without effector molecule, it's not possible to synthesize the secondary messenger. If we want to synthesize the secondary messenger, compulsory we have to activate the effector molecule. Once secondary messenger activated, then the signal will come inside. It will take the information till the nucleus level. Then it will allow the gene to get expressed. It will allow the gene to get transcribed. Simple point. Now everyone pay attention towards the diagram. What happened, friends? Initially the GPCR is not activated. Why? The ligand is not present. When the GPCR is not activated, which is also not getting activated, G protein is also not getting activated. Simple point. Now when the ligand come and attach to the GPCR get immediately activated. Now to the cytosolic domain of the GPCR, who will come and attach us? Very simple point. The G protein alpha will come and attach us. Very simple. The moment G protein alpha will come and attach us, G protein alpha converted from GDP back to GTP. I will explain how. By an important protein or important exchange factor, which everyone has to remember. What do we call that factor? We read many times. Guanine exchange factor. We shortly call it as GEF. What the GEF is going to do? It is going to convert G protein alpha GDP form into G protein alpha GTP form. Now everyone has to listen. The moment, understand friends, understand, the moment G protein alpha convert from GDP to GTP form, it lose its association. What, friends? It lose its association with its friends. What are the friends? Beta and gamma. Immediately what is going to happen? Once it lose their association with beta and gamma, alpha come out. I already told you very clearly what is the ultimate goal of a G protein. Ultimate goal of the G protein is what, friends? See here, activating the effector molecule. So you have to listen the statement carefully. Z protein alpha can activate the effector molecule only whenever it is in the GTP bounded form. If it is not in the GTP bounded form, it's highly impossible. It cannot activate at all. Now every one of you listen my statements very carefully. Now immediately what happened? The effector molecule, I already told you, effector molecule is the one which is involved in the activation of secondary messengers. Simple point. Now what the secondary messengers are going to do? They are the one which is taking the information till the nucleus level. Anyway, we will read secondary messengers very briefly and elaboratively by taking every single secondary messenger as an example. Now this is an important question. I have already told you, friends. Here you only try to understand, friends. How does the GPCR get activated? Of course, because of the ligand. How does the G protein get activated? Of course, because of the ligand. How does the effector molecule get activated? Of course, because of the ligand. How does the secondary messenger get activated? Of course, because of the ligand. You can call ligand, or you can also call it as a primary messenger. Anything is same. Sometimes instead of giving a name ligand, they can call it as a primary messenger. You can call primary messenger or you can call ligand. There is no doubt about it. There is no difference. Now listen everyone carefully. What are the various kinds of ligands that use the GPCR? Everyone listen to me, my friends. Please listen the statement. Depending on the ligand, we can differentiate the GPCR into different types. Listen my statements. Depending on the ligand, we can differentiate the GPCR into different types. Depending on the ligand, there are different type of effector molecules are there. Depending on the ligand, different type of secondary messengers get activated. Depending on the ligand, different response can be shown. In a simple way, I can say to all of you, everything is exclusively depends on the ligand. Now my friends, let's try to understand what kind of ligands that I am explaining very elaboratively and briefly. By that we cannot miss any other point when it comes to the point of exam. So pay attention, we don't skip anything. Now what kind of ligands are going to use, because this concept is very important. We should not leave a small point also. Now listen, let's take an example of epinephrine. Friends, epinephrine is a ligand. Now I told you it is going to use which receptor? Of course, GPCR receptor. Now these are all GPCR, no doubt my friends, these are all GPCR, but the names are different. Why we have given the different names? That's what, based on the type of ligand that it is associated. This epinephrine associated with a receptor called as beta adrenergic receptor. Never forget the statement. What receptor? Beta adrenergic receptor. It's a type of GPCR only. So epinephrine will come and attaches to the beta adrenergic receptor. Ultimately we have to activate what I told you, everything very clearly. We have to activate the effector. What is the effector involved here? My friends, the effector involved here is the adenylyl cyclase. What the effector molecule is going to do now? They are going to activate the secondary messenger. We will read later. Now ultimately what the secondary messengers are going to do? They are going to show a particular response. What is the response here? Breakdown of the glycogen into glucose. That's the physiological response. Now let's take another example. Take an example, acetylcholine. Acetylcholine is also ligand. It is also using what? GPCR only, no doubt about it. But here what is the name of the GPCR receptor? You can call it as a muscarinic receptor. Do not forget the statement at all. Now what is the name of the effector molecule? Here the effector molecule is not adenylyl cyclase. You can ask me why? Because here the signal is not epinephrine, it is acetylcholine. What is the point you have to remember now? Based on the ligand, everything is going to be changed. Based on the ligand, receptor will change. Based on the ligand, effector will change. Ultimately, based on the ligand, the function or a response also going to be changed. So the overall point you have to remember, the type of secondary messenger, the type of effector, the type of receptor are going to get activated and going to show their response exclusively based on the ligand. This is the point I want every one of you remember. Never ever forget this particular point. Here comes another unique statement, friends. What is that unique statement? What are the type of ligands uses this? I am requesting all of you, whatever I am marking it, please kindly remember that. If you remember our 2019 question paper, we will solve it. The 4 marker question was directly asked. Simple question for 4 marker. Type of ligands they have given in one column, and they have given in other column they have asked whatever the type of receptors that they are going to use. Simple. Now listen to me very carefully. Type of neurotransmitter, whatever I am marking, please remember without fail. Acetylcholine, dopamine, noradrenaline, GABA. 4. Glutamate, 5-hydroxy tryptamine, MSH, melanocyte stimulating hormone. Then somatostatin. My friends, I am requesting all of you to remember this 9 neurotransmitter without fail. They are all utilizing the GPCR as their major receptor to show their action. Now if you come to the hormones, remember what I am marking. Adrenaline, angiotensin, glucagon, histamine, melatonin, prostaglandins, vasopressin. Please remember this is 7 without fail. These 7 uses what? These are all very important one, means oftenly or repeatedly asked in the exam. These are the hormones and these are all the neurotransmitter in order to show their action exclusively depending on the GPCR. Okay, now listen to me very carefully. Let me quickly summarize this whatever we read now. What we talk about the G proteins. G proteins also known as guanine nucleotide binding proteins. They are a family of protein that act as a molecular switches inside the cell. Why the G proteins are required? They are involved in showing a variety of responses. See friends, the same direct statement asked in the 4 marker question. Direct statement. What is that? Z proteins belongs to the larger group of enzymes called as GTpases. We read very clearly. My friends, listen this paragraph. Look into this. Z proteins are commonly used in the signal transduction pathways in animal cell in response to what? Diverse stimulus such as light. So if we want to see, we have to use the GPCR. If we want to smell, we want to use the GPCR receptor. For hormonal action, for neurotransmitter, for chemoattractants, everything exclusively depends on the Z protein only. My friends, please kindly don't forget the statement. They consist of what, friends? Heterotrimers of alpha subunit, beta subunit, and my friends, followed by the gamma subunit. Don't forget the statement. Okay, now listen to me very carefully. What are the types of G protein? Friends, there are different type of G proteins are there. In the last PPT, I have mentioned very few types of G protein. Here I have mentioned many. I am requesting all of you to remember all. One we called G protein S. My friends, S represents stimulatory. S represents stimulatory. I represents inhibitory. These two things you people remember. Now you can ask me, sir, what is this stimulatory? What is this inhibitory? Now you can also get a doubt that when the G protein stimulatory can be utilized, when the G protein inhibitory can be utilized. Everyone has to listen my point. Sir, it depends on what? When the G protein stimulatory only comes into the picture, when the G protein inhibitory comes into the picture, it depends on the type of ligand. Everything is depends on the signal or the ligand molecule. So you only try to understand when the G protein stimulatory comes into the picture. When, listen friends, whenever it want to activate the adenylyl cyclase, always keep that point in your mind. Whenever it want to activate the adenylyl cyclase, with respect to what? With respect to a particular ligand. Same, you see here G protein olfactory, OLF. When this G protein olfactory comes into the picture, whenever they want to activate the adenylyl cyclase present in the sensory neuron, then only G protein olfactory will come. Now, when the G protein stimulatory come and olfactory will come, of course it depends on the ligand. Everything is depends on the ligand. Suppose I want to activate the adenylyl cyclase present in the sensory neuron, then a particular ligand will come and attaches, then only G protein olfactory will get activated. Suppose I don't want to activate the adenylyl cyclase at all. Now what should I do? I have to inhibit the adenylyl cyclase. My friends, if I want to inhibit the adenylyl cyclase, what should I do? We have to use the G protein I. I represents inhibitory. Stimulatory activated adenylyl cyclase, olfactory activated adenylyl cyclase, inhibitory what it will do? It will inhibit the adenylyl cyclase. It is also depends on what? Undoubtedly, my friends, it is also depends on the ligand. Never ever forget that particular point. Now my friends, I am requesting all of you remember this thing especially. G protein T. What is T, friends? T we call transducin. What is the speciality of this particular transducin? Now the transducin is not going to act on the adenylyl cyclase. It is not going to act on the potassium channel. What is the goal of this G protein transducin? It acts on a effector molecule. What is that? Cyclic GMP phosphodiesterase. Now you can ask it, you can get it out. Why the G protein transducin is only act on the cyclic GMP phosphodiesterase? Of course, that is also exclusively depends on what, my friends? Undoubtedly based on the ligand. Now very unique one. G protein Q. Q represents quotient. I am requesting you, my friends, I want you people to remember all the information. With great difficulty, I am summarizing all. By that which you cannot miss any other, we cannot miss any important points. Focus. Now G protein quotient, it is going to activate not the adenylyl cyclase, not the cyclic GMP phosphodiesterase. It is acting on a specialized effector molecule. What is that effector molecule? We can call it phospholipase C beta. So friends, what I can summarize to all of you, I can summarize a point that what kind of effector molecule to be activated? Specific type of G protein only will come and activate that effector molecule. Now, what kind of G protein has to come into the picture? It is exclusively depends on what? It is exclusively depends on the ligand molecule. Please kindly do not forget this particular statement at all. Right. Friends, is that clear everyone till this particular slide? Hope everyone is clear with this particular slide. Friends, that's all for today, and please kindly listen my words. Day after tomorrow, the same class will be continued. Day after tomorrow. The class will start from 7. Please keep it in your mind, friends. Class will start from 7. Class is not going to start from the 7:30. Okay. Day after tomorrow, it is 7 to 8:30 p.m. See, 90% I will try to finish the entire cell signaling. At least 90% I will cover, which is important. And friends, what about the tomorrow class? Tomorrow in an academy, I am going to take one particular class on the Omicron. How dangerous the Omicron variant is. How dangerous the Omicron variant is. That thing I'm going to explain you in the Unacademy lecture. Everything about the Omicron, their molecular mechanism, how many number of cases are identified so far, what is their structures of protein spikes, everything we will understand elaboratively. And the link for this YouTube class I will share you in the Telegram. This will start tomorrow 7:30 p.m. Okay, and we are going to continue the cell signaling day after tomorrow 7 to 8:30 p.m. 7 o'clock class, and we're gonna finish it. Please kindly don't miss any class. Okay, thank you so much, friends. Thank you very much. Thank you. Good night. Bye bye. Good night.